here, and let's go ahead and get started on this. Let me shut the door so we don't get contaminated by chemistry. Oh. Okay. Today we're going to take a look at the special senses, the eye, the nose, and the mouth, the ability to taste and to smell and to see. And reminder that the muscle test has opened up. It will be open until this weekend. I will likely extend it one day for you. I'm going to send out a list. You already have a list of all the muscles. I will send out an email just trying to emphasize what you should review for this test. There are only 60 muscles involved. Many of them are clustered together like the quads and the hamstrings and the rotator cuff, and things like that. And just a reminder that AP2 in May term is open for registration. It will be a hybrid class, the same format we're having for AP1 lecture and lab right now. So you don't have to attend in person. So, okay, having said all that, let's go into special senses. Okay, special senses. There's only 5 of them: smell, taste, vision, hearing, and balance. That's equilibrium. They are all located in the head. They are all found in very sophisticated sensory organs: the eye, the ear, the nose, the mouth, and the inner ear for balance. They have a very sophisticated nerve pathway that they have to follow. They are not free nerve endings, not just the open end, the exposed end of a dendrite. They are, and they're not scattered throughout the body. They're all located in our head. The first one we come to is olfaction, the sense of smell. Now, this takes place inside our nasal cavity at the anterior surface, meaning the top surface of the nasal cavity. It would be in this region. Put this up here. Friend out of the way. And put the camera up. Right. Oh, very good. This is the anterior surface of the nasal cavity, located right here. And this is the only area where our olfactory receptors are located, in this region here. The rest of the nasal cavity is lined with mucous membrane and ciliated structures to move the mucous membrane around. But this area right in here is where our olfactory mucosa is located. So our sense of smell depends on stimulating the receptors there. Now, for us to be able to smell anything, let me get out of this. Okay, now back to this. Every odor is based, that we detect, is based on molecules of chemicals in the air. Now, for us to smell anything, those molecules have to land on olfactory receptors. And to do that, they have to dissolve in mucus that coats the inside of our nasal cavity. So they have to be in a fluid medium so they can absorb onto the receptors. And we have about 100 million olfactory epithelial receptors located there. And they are, each receptor has a bipolar neuron that will send the signal to the central nervous system if we can hit threshold. We have the ability to detect about 10,000 different odors. Now, what will happen is a molecule will drift into the air, dissolve in the mucus, come in contact with the olfactory receptor, a signal will be sent up the various branches of the the olfactory nerve, up through the cribriform plate of the ethmoid bone. That's a perforated plate right above the nasal cavity. And then it will pass up to an area known as the olfactory bulb. From there, all these signals will be collected in the bulb, and now it will proceed downwards, bless you, known as the olfactory tract, because now we've moved into the central nervous system. We've gone from a nerve to a tract. And so we process this. Again, though, the receptors in our nose don't have any clue what they're detecting. They're being stimulated. Our brain has to interpret these action potentials coming in from the bulb, the olfactory bulb, so telling us, what is it? Is it a rose? Is it baking bread? Is it chocolate in various forms? Is it, garbage? Is it, a dead animal? Is it the smelly drains that were backed up at the other end of the hall earlier today? That's what the problem was. So, so our ability to detect that is based on our brain remembering these odors and identifying it. Like natural gas that you cook with and heat your house with. If it's natural gas and not, if it's methane, it has what we call an odorant added to it to stink, because natural gas by itself occurs naturally. That's what I call it, natural gas. It's odorless. You have to add a stenching agent to it to make it smell bad. So it alerts you that you have a gas leak in your house. And then so that way you can light a match to see where the leak is. You only do that once. Anyway, we can detect about 10,000 different odors. We can tell the difference between roses and hyacinths and maybe tulips. We can tell the difference in odors between baking bread and grilling burgers or something like that. We have all these different memories here. Olfaction also helps us to eat. The ability to detect odors also helps us with our detection of food flavors. And one of the consequences of aging is that we gradually lose up to half of our olfactory receptors and half of our taste buds, our gustatory receptors. And so in your elderly patient, they may be suffering from malnutrition because food doesn't taste like it used to. That's their complaint, because most of their taste buds and olfactory receptors aren't functional anymore. And so they don't have any flavor. And so they don't perceive it as, they lose interest in eating. And so they don't eat. Or when they do eat, they overload it with salt or seasonings to try and get some flavor back. Or they will overload, they're not aware of odors that they may be releasing. They're not aware of, did they shower for the past week? They smell funny? Or perhaps they put on a little too much aftershave before they go to church. And they wonder why nobody in 3 pews in any direction are not sitting around it, because there's this cloud of men's cologne sort of laying there. Yeah. So and it's not anything, it's not their fault in the sense that they can't tell anymore, So now this is what it looks like, more or less. Here's our nasal cavity right here. The cribriform plate is located of the ethmoid bone is, if you remember from bones, is located right here, right above the top of the nasal cavity. You remember, it's perforated so that branches of the olfactory nerve can go through there. The olfactory nerve then moves into a larger area here called the olfactory bulb. And in us, it's pretty small. Next week, when we look at the sheep brain, you'll see that sheep have a pretty large olfactory bulb, because sheep depend on a sense of smell a whole lot more than we do. But the olfactory bulb allows us to collect all of our input from the olfactory nerves, the branches of the olfactory nerve, and then we send that down the olfactory tract. And the area in the brain processes it is probably over here on the side somewhere. We'll talk about that in lecture when we get into chapter 12. And so we process the information so we can be aware of what the odor is. We may not recognize it as an odor we know. We may recognize it as a brand new odor, and then we'll store it in our memory. Now, what the olfactory membranes look like, here we see these blue structures are the bipolar neurons right here. These are the bipolar neurons. They are they have an axon. They have an axon and a dendrite. Down here's the dendrite end. This is the axon end up here. And here are your receptors located here. And you're going to, if you hit threshold, you'll send a signal up these various axons into the olfactory nerve up here, which will then synapse in the olfactory bulb and move into the olfactory tract. A lot of olfaction going on here. As I said in lecture, only the olfactory nerve and the optic nerve are going to use these bipolar neurons. It's a very efficient way of getting information moving along here, getting action potentials to move. So some of this and the retina are probably the most complex of the sense organs that we encounter. So now taste occurs, well, the sensory receptors are located in our mouth, on our tongue. We also have sensory receptors on the epiglottis. The epiglottis is a structure that we use to close off our airway when we swallow. We have taste buds scattered throughout the tongue and on the epiglottis. And again, we depend on these chemicals, these chemical agents in the food to dissolve in saliva and then spread across the surface. Now, how many tastes can we detect? Well, we used to say up until the mid-'80s that it was sweet, sour, salty, and bitter. And there was even a misinterpretation of some of the early work done on taste buds by saying there were specific regions on the tongue that only had sweet and sour and salty and bitter receptors. And if you think about that, that's not logical, because if you eat something that's bitter, you're not going to, you taste it, you're not going to taste anything if it's not, according to this logic, that if it's sweet at the tip of the tongue, something that's bitter isn't going to be detected until it gets in the side of your mouth. Or something that's salty might not be detected until you've already eaten part of it.
So how do we even rationalize that? I always wondered that. Like, how do we ever get to a point where we came up with that as a theory, but then we agreed upon it, even though it would be as simple as take a grain of sugar or something else, put it on the wrong spot of the tongue, and the text should be debunked right then and there.
Exactly. And the basis for that was it was a German physiologist about 100 years ago that detected all these different regions, these different sensations, and his paper was misinterpreted. It was translated wrong. And so for well over about 100 years, everybody was saying just that: the tip, the sides, the back, the middle, all had different sensations.
Well, even growing up, I saw diagrams like that.
Oh yeah, of course. It's only been in the past 10 years that information finally got corrected. And there are still people that insist, and you'll find it in textbooks, that insist that the old interpretation is true. But it doesn't make any sense, because how can you have an area of the tongue that is specific just for one flavor when you're going to be putting all the flavors together anyway? So, because every taste bud will be responding to a different sensation, and your brain can do the interpretation. So, and I think a lot of it had to do when a fifth taste came along, a fifth taste called Yumami. It is a savory flavor is what you enjoy when you eat protein, like grilled chicken or burgers or steaks or anything like that. That special flavor that you get. And while chicken and burgers and steaks all taste different, the grilling sensation, the savory flavor of the protein in there is pretty consistent. And that has become the fifth taste sensation that has been described. So we can sense that too. So the early work, the work that was done 100 years ago was very valid, but it was done in German. And when it was translated incorrectly. And even on this campus, even at Walter State, there are faculty that insist that the tongue still has its regions. So I don't argue. I just teach it the right way, that's all. So anyway, now, so these are the areas in the tongue. The taste buds are they're chemoreceptors. Just like in the nasal cavity, they are chemoreceptors. They detect changes in chemicals. And so they respond to different types of chemicals. The taste bud is a little less sophisticated than the olfactory receptor, but it doesn't matter. The taste buds are located on our tongue, on the roof of the mouth. That would be the soft palate of the roof of the mouth, and along the epiglottis. When we get into AP2, we'll talk a lot about the structures of the nasal cavity and the oral cavity. But the soft palate is the area near the back of our oral cavity, just in front of the uvula. The uvula is a little thing that dangles down in the back of your throat. So the soft palate is just anterior to that. So we can pick up all these different odors. The taste buds themselves, the papillae, the taste buds are found in these raised bumps called papillae, and the papillae look something like this. And down inside the papillae are the taste buds, and they look sort of like this, I don't know, like a turnip, if you will, or something. But we use, they're going to be using the unipolar neurons, since most of our sensory reception, other than the eye and the nose, use unipolar, and they wear out too. We have thousands and thousands of these taste buds, but they wear out over time. And an older person, an elderly person may tell you something like I said, that food doesn't taste the same anymore. That they'll say, well, I used to eat Cheerios for breakfast for years, but they don't taste right anymore. And yet you take a 5-year-old and you give them Cheerios. The most children around 5 years old have the most sensitive taste buds and olfactory receptors. You can't fool a 5-year-old with knockoff Cheerios. They know the difference. You can't give them fake Oreos because they don't taste the same. We all know that, they don't taste the same. And so children can easily detect the difference in sensations. Most of us remember, have our favorite flavors, and we expect our food to taste that way. And I would imagine it's probably pretty disappointing when you're dealing with, if you have an elderly patient who can't remember can't detect these odors and flavors anymore. They're expecting it to taste a certain way and they don't get anything. It would probably be pretty disappointing. And you'll see that a lot of cases of malnutrition in your elderly patient. A lot of it doesn't have to do with lack of resources. A lot of it also has to do with they lose interest in eating. Yes.
This is a really weird question.
Okay.
Can you pull off a taste bud? Because I tried to before when I was younger and it didn't work.
It would be difficult. I would imagine you could try it.
I really wanted to pull one off and look at it under a microscope.
Okay.
I tried. I could not pull it off.
They are very tiny. That's the problem. They are about the size of a pore on your skin. So they're extraordinarily hard to find. Just because you can feel the bumps on your tongue, the taste buds are inside that. So it's possible, but it would be very challenging. So, okay, so news at 8. So now, so that's our ability to sense odors and to taste food. And in the past, we've done interesting things about, there's interesting relationships between the taste bud and the nose. We've done this pre-COVID. We used to do something like this, where if you take somebody, a volunteer, and you blindfold them, and you take a piece of raw potato and a piece of freshly cut apple. And if I were to hold the apple under your nose, but give you the raw potato, now the texture of a raw potato is very, very similar to a fresh apple. You'll smell the apple and you'll eat the potato, and you will swear that you're eating apple. Because the odor. Because remember, don't look at me like that. No, it really does work, because the odor of food goes a long way in our perception of taste. Consider how you feel when you have a sinus infection and you're all clogged up, and you're talking like this, and you can't breathe, and you got a lot of congestion, and you try and eat something and it doesn't taste very good. You're reduced to eating chicken soup because it's already bland, and you can't taste anything or taste things very well, because you're not smelling them. You're not smelling the food. So the odors of the food go a long way in determining our ability to sense what it is. And the only reason that you would eat the potato is because you wouldn't see that it was a piece of potato. It wouldn't look quite the same as an apple, but it sure has the texture and the feel of an apple. And if you smell the apple, you would taste the apple in the potato. I even thought about trying with a French fry, giving somebody to eat a French fry at the same time they're smelling an apple. They're like, well, it feels like a French fry, but it tastes like an apple. Is it an apple fry? I never tried it, but I wanted to. So there's a lot of things you can do. If we ever get back to normal, we might do stuff like that again, but right now, who knows? Okay, moving on to the eye. The eyeball is probably the most complex of the sensory organs that we have. It's located in the socket, the eye orbit. The eyeball itself is about the size of a walnut, and it has a series of 6 muscles around it to move the eye around and to control not just the movement of your head, but that's a separate set of muscles, but the movement of the eye within the socket. It is innervated by the optic nerve, and the optic nerve passes through the opening in the optic canal or optic foramen, going back into the brain proper. And next week we'll take a look at the structures that show how what the optic nerve does to get into the brain. The outer covering of our eye is very specific. It's called the sclera. It is the white part of the eye. It is white all the way around. And if I can get this to open up, there we go. So the white of the eye is the outer covering of the eyeball, like what we have here. Let me show this to everybody. Okay, now this is a rather large eyeball right here, and this white area here is the sclera, the outer covering, and then covering the sclera is a layer called the conjunctiva. It's connective tissue that protects the sclera and it covers the sclera all the way up to the layer of the cornea. The cornea is the transparent area in the front of the eye. The cornea is this transparent region that you see right there. So the conjunctiva covers all the way around on the visible portion of the white of the eye of the sclera, and except this area where the cornea is. If you've ever had conjunctivitis, also known as pink eye, a lot of us have had pink eye at some point or another. You know, go to a swimming pool in the summertime with an infected kid and you're going to come home with pink eye, likely. Pink eye is that condition where you're overnight your eyelids sort of glue together, you know, and it's very uncomfortable. And you take antibiotics and it goes away. It's an inflammation of this layer around here. So there's our cornea. The cornea is a transparent layer, transparent structure in the front of the eyeball. And let's see, let's go back to this. Now, light rays come in through the cornea. They pass through an opening called the pupil. The pupil is a hole in a structure known as the iris. The iris is the area of the eye that is pigmented. It gives us our color, whether we have blue eyes or brown eyes or green eyes or hazel eyes or black, whatever color, you know, or black, brown, green, hazel, blue, gray, whatever combination, whatever permutation of eye color you have, that is the iris. And the iris then is a set of smooth muscles that will open and close to create the pupil. Let me show you what that looks like here. Now this is the iris. In this case, this is a brown eye. The iris is pigmented brown. And you can see this opening here is the pupil, and it can enlarge, it can expand, it can contract. We have two sets of muscles in the iris that will allow the pupil to enlarge to let in more light, or constrict to let in less light, because we want to regulate the amount of light that's going to be coming through and ultimately hitting the retina in the back of the eyeball. Okay, so and there's a cross section of our eyeball here, and the eyeballs we'll look at next week in lab are very, very, very similar. They are either cow or sheep eyeballs, and they are practically identical to humans. There's only one difference, and we'll show you that later. So this is our eyeball. The outer coating, outer layer, the white of the eye is the sclera. There's a middle layer called the choroid coat, and the innermost layer is called the retina. Now in the front of the eye, we have, you can see where the sclera comes all the way down to the cornea. The conjunctiva covers the sclera everywhere that's visible to the outside in the socket, except or in the orbit, except where the cornea is. Light rays come in through the cornea. They are slightly bent there. They enter the eyeball and through the pupil. They pass through the lens, and they are projected onto the retina themselves. The retina is where the photoreceptors are located. There's two key structures on the retina. We have an area called the fovea centralis. That is the area of greatest visual acuity. The other area is called the blind spot. That's where the optic nerve leaves the eyeball. There are no photoreceptors on the blind spot. So light rays that strike the blind spot don't do anything for us. There's nothing there to stimulate them. Now the lens is held in place by two structures. It's held in place by what's known as the suspensory ligament and the ciliary body. The ciliary body allows the muscle, our muscles that allow the lens to change its shape. Our lens, while it looks like it's a pretty rigid structure, it looks a lot like this. Let me put it up here real quick. This is the shape of the lens right here, and the lens sets inside just behind the iris. The lens pretty much looks like this. It is a lot smaller, of course, and when we take out a lens from an eyeball, you'll see it's about the size and shape of a M&M, a transparent M&M. So, but it isn't, what we take out of the cow eye is going to be a very rigid structure, but in reality, it has a little flexibility to it because we have this ability called accommodation, where we can change the shape of our eyeball, of our lens, to look at things close up or to look at things further away. Now let's move on here. The ability to see things depends on light rays coming into our eye. Everything is given off light rays. My shirt is perceived as blue because blue light rays are bouncing off my shirt. Yours is purple, yours is gray, yours is whatever, yours is yellow, okay? Those are the light rays bouncing off. Well, yours is multicolored, so you have a lot of light rays coming off. Light rays bounce off of every object. They strike our eyes. The lens allows those light rays to be focused on the retina. And when they're focused on the retina, they're going to activate photoreceptors. And so depending on what we're looking at, we can change the shape of our lens to focus on objects close up, or we can change it to look at something in the distance. And we're not even aware of this. This is called accommodation. The ciliary muscle that surrounds, you'll see it when we take the eyeball apart next week, is a ring of muscle that's right behind the iris, and it controls the shape of the lens. The light rays are traveling in packets called photons now, and that's, okay, light rays, we'll just let it go at that. The photoreceptors, though, are called the cones and the rods. Now, an easy way to remember which is that cones are color receptors. Cones are color receptors. C for cone, C for color. They give us the greatest visual acuity. Cones are color receptors. They give us the greatest visual acuity. They let us perceive things in colors, and we get our sharpest vision from the cones. The fovea centralis, the area of greatest visual acuity, is nothing but cones on the retina. So when we hit that spot with light rays, then we are seeing to the best of our ability. The rods allow us to see in dim light. They also, because they are found on the edges of the retina, allow us to see with peripheral vision. The object moving in the corner of our eye. The dog that runs out in front of us in traffic, and we can just catch a glimpse of it before we try to stop so we don't hit it, or the kid, or whatever. It's all about priorities. Anyway, the corner of our eye vision, we don't really have corners in our eyes, but the peripheral vision is very sensitive because the edges of our retina are packed with lots and lots of rods. Dim light allows us, dim light requires the use of the rods. We don't have good color vision in dim light. At night, our color vision is shot because there's not enough light out. But we can still see things because of our rods. Our rods give us night vision, not night vision like night vision goggles, but at least we can see in the dark. We can see to some extent. So two different types of photoreceptors, two different purposes. Both work very effective for us, are very effective for us now. So anyway, I said about the iris. The iris, the pigmented region of our eyeball, genetically determined. Blonde hair and blue eyes, for example, are recessive traits. They usually go together. If you have blonde hair, you're likely going to have blue eyes. It's a genetic recessive trait. It's the pigmentation. Doesn't mean anything special. It's the luck of the genetic draw. So whatever color your eyes is. Doesn't help speaking. So whatever color the iris becomes after when you're born, let's put it that way, is genetically determined by your parents. But what the iris really does for us is open and shut the opening of the pupil. We only need a certain amount of light to strike our retina. Too much light is unpleasant. If you were to shine a flashlight into your eye to see what would happen, it would be depending on the intensity of the flashlight, it would be pretty uncomfortable. You wouldn't like it. And your pupil would shrink into a very tiny opening. Try and minimize the amount of light striking there. On the other hand, if I were to turn the lights out in the classroom and we were sitting in the dark, then everybody's pupils would enlarge. Try and maximize the amount of light coming in. We're always trying to adjust to get the maximum amount of light that we need, not what we can take, but what we need. We don't, yes?
Why does the iris change color sometimes? Like when you were born, it can be like blue and then turn brown and stay brown.
Good question. It is what we call incomplete dominance. It is some genes, when the genes you have, if you have two sets of dominant genes, which you can easily do, like in eye color, they will compete with each other. And sometimes the gene for brown eyes wins out. Brown is always a dominant gene anyway. And blue eyes are always a recessive gene. So if you were born with blue eyes and they eventually turn brown, it's because the dominant gene, the brown gene, produced more pigment for the brown color. Some people have hazel eyes, which is a mixture of green and brown, both dominant. But you have hazel. People say that their eyes go from gray to blue, and because it's how the light hitting it. It's also the expression of the genes. Consider I have a basset hound with brown and black and white markings on her. So incomplete dominance of each color. So cows, cows aren't all black or brown or white. A lot of them have white heads and they have black bodies, and some are brown. Some have yellow markings on them. So incomplete dominance again. And so it depends on when it gets expressed. So believe it or not, I had blonde hair when I was a kid, and it has gradually turned brown over time. Yeah. Blue eyes haven't changed, but blonde hair disappeared. It turned brown. Was yours lighter when you were a kid?
It was always just kind of dirty blonde.
Okay. Yeah.
Maybe a little bit lighter. So might.
Mine was super blonde when I was little and now it's brown.
And it happens. I don't know if it was. I mean, I grew up in Florida and I was out in the sun a lot. But when I was a kid and we moved to Pennsylvania, it started turning brown, and went through all the dirty blonde to brown stages. And, again, partial dominance, because the blue eyes and the blonde hair are always recessive traits, and they can be overwhelmed by the dominant traits. And if you have any dominant traits still hanging around, because we're always a mixture, that's what happens. Okay. So so we have the iris in front, and we'll discover that with our cows, most of our cows have brown eyes, because we can take their iris out of the eye, and we'll see the ciliary body behind that, and then we'll see the lens behind that. And the lens will help focus the light rays coming through onto the retina. Okay. The retina is where our photoreceptors are located, the rods and cones. The center of the retina is the fovea centralis, where we have nothing but cones. They are where we have our greatest visual acuity. This is where when you're reading something and you say, I need to turn the light on so I can see it better. And the light goes on, and now you can see it clearly because you are stimulating the cones in the fovea centralis, the center of the retina. Nothing in there, nothing at that location except cones. So you have your greatest visual acuity right there. So now this is a retina. This is our retina. Our retina is built backwards, or at least it seems that way. You can see which way we're looking at the center of the eyeball is where the light's coming from. The light's coming from the lens. It's going through the eyeball itself, and it hits the retina. The retina is on the back wall of the inside back wall of the eyeball. This yellow layer here is the retina, and you can see right here. There's the sclera, there's the choroid coat in red, and there's our retina right there. So what we're looking at on that slide is a cross section of that very thin retinal layer. Light comes in, and the photoreceptors that you would expect to be right here in the surface are not there. The photoreceptors are down here. So there's the retina, there's the choroid coat, and there is the sclera right there. Now this layer right here are the ganglion neurons. What that means is those are the axons leading to the optic nerve. They're on the surface of the retina. Underneath that, you have the bipolar neurons, the one with the one axon, one dendrite. One end, the axon leads to the optic nerve, and the dendrites are going to receive information from the sensory receptors. And the sensory receptors, the photoreceptors, are here. The rods and cones are butted up against the choroid layer. So it seems like it would be just the opposite of how you would expect it to look. The rods and cones are deep inside the layer of the retina. They're on the inside layer of the retina. This actually works to their advantage because the rods and cones heat up as they become stimulated. And the heat energy is dissipated as the heat is generated. They are dissipated into the choroid layer, and instead of heating up, if they were on the surface of the ganglion, they would heat up the ganglion and possibly damage the optic nerve. So it's sort of an unusual, it seems like it's a backward arrangement. It doesn't really, it's not, no one's ever going to hook and say, hey, your eyeball's backwards, but it's just the way that the structure is located. The bipolar area is in the middle. The rods and cones then are up against the middle layer of the eyeball. So the rods and cones are named that because of their shape. The rods, the dim light receptors, are shaped like a big pole or a rod. The cone looks somewhat like an ice cream cone. The rods allow us to detect images in very dim light. They use a pigment, a visual pigment known as rhodopsin. Rhodopsin is derived from vitamin A. And when we were all kids, we were probably said, eat your carrots so that you have good eyes. Vitamin A is derived from carotene, found in the orange pigment in carrots and in orange juice and in our vitamins. And so the more rhodopsin we have, the better, the greater the greater functionality we'll have in our rods. You know, a person who is experiencing night blindness may have an absence of vitamin A in their diet. So the cones are all about color vision. They pick up reds and greens and blues. A person who is colorblind has a deficiency in one set or maybe two sets of those rods. The most common type of colorblindness is red-green colorblindness. So now let's take a look. Here's what the rods and cones look like. This is a rod over here on the left. The rod is shaped. This is the rod-shaped structure here. And over here is the cone here. And the arrangement in the retina, you can see there's the ganglion cells that are going to connect to the optic nerve. Here are the bipolar neurons here. The dendrite end of the bipolar neuron connects to the rods and to the rod or to the cone, and the axon end connects to the ganglion. You know, light's coming in, you know, this way from the ganglion. That's the edge of the retina right there. Going to go past through that. Going to go past the bipolar neurons. Going to go down to the rods and cones and stimulate them at this end here, all the way in here. So it does seem like it's a backwards type of arrangement, but it really isn't, because it works very well. So as I said, the rods tend to be grouped more along the outside edges of the retina, and the cones are centered in the middle. And the rods are intermingled, but when you get into the fovea centralis, it's nothing but cones. So, yeah. We find that, and this is a phenomenon that we've all seen, if you're in a dark room and you go into a brightly lit room, you can see very well. I mean, of course, we're all going to go, oh, you know, as our pupils constrict. We've all had that, right? You know, so our pupils constrict, but we can see very well when we go from a dark place to a bright space. We can see very clearly. However, if we go from a bright space to a dark space, it takes longer to accommodate. It takes longer for us to adjust. The cones will very quickly become activated when we go from a dimly lit space to a bright space. The cones adapt very quickly. The rods take time. The rods take time to generate rhodopsin so the rods can be efficient. So if we're outside in the dark and someone shines a light in our face, our night vision is gone, and it'll stay gone for about a half an hour. You know, our ability to perceive things very well in the dark, well, it'll recover, but it takes time. You know, we've destroyed that rhodopsin with the bright light. It will take time for that rhodopsin to come back. So, and then once it comes back, then we can see well again in the dark. But you know that when you go from a brightly lit space you go from a bright room into a very dark space without turning the light on, you can't see very well in the dark. Given time, everything seems very clear. You can see very well at night if there's even a little bit of light. So our pupils help with that. The iris will open and close the pupil as needed. You know, when we are in bright light like we are right now, the iris is going to be constricted to limit the amount of light coming into the retina. We don't need a lot of light for our retina to work just fine. If we were outside where the light wouldn't be as bright as it is in here, our pupils would be slightly enlarged, what we call normal outside light. You know, again, we don't need to have them wide open. We don't need to have them constricted. And yet, if we were to turn all the lights out and close the blinds, it'd be very dim in here, then your pupils would accommodate by getting larger. The iris would constrict to allow the opening to get bigger. We have to, because the reason we do that is we're trying to get the same amount of light to strike the retina all the time. So we're accommodating for the intensity of the light, whether it's dim light or bright light or what they call normal light. Define normal, who knows? But we can, our pupils will adjust to that. Now, the blind spot is an interesting phenomena here. We've probably all heard about a blind spot, you know. There are no photoreceptors on the blind spot. And so when light rays strike there, nothing happens. There's no stimulus, there's no action potential, there's no nothing. There's a little test that we do when we're all on the ground. A little card I have that I give out to everybody, and has a plus and a dark spot on there. And it's a very simple test for detecting a blind spot. You simply take the card and, for example, you would take the card and look at it, shut your left eye and focus your right eye on the plus, and then bring the card in toward you. At some point, the light rays from the black spot are going to strike the blind spot in your retina. And what's going to happen? You're not going to see it. The blind spot, it doesn't have any photoreceptors. So when the light rays coming off of the black dot hit the optic nerve where it's leaving the eyeball, it disappears. You're bringing it in and you're bringing it in. All of a sudden, it disappears out of your field of view. You can't see it anymore. It's still on the paper, but you can't see it at some point when you're bringing that card into your face, because the light rays bouncing off that dot are hitting the blind spot in your eye. And then you can turn around and do it with the other side, and it works just as well. And this is one of those very simple little tests, but it works very well to show you that there really is a blind spot. We've all experienced it. Sometimes you're driving down the road and you look, you don't see anything, and you start to go and then you stop suddenly because where'd that car come from? You know, because it's like just for a split second, as we'd like to say, the light rays coming off that car that you didn't see were hitting your blind spot. A millisecond later, it's there. We've all experienced that. Okay, inside the eyeball, it isn't just a hollow cavity. The back part of the eyeball is filled with jelly. The front is filled with water. The eyeball is divided into two parts at the lens, and the area in front of the lens is what we call the anterior chamber. The area behind the lens is the posterior chamber, and the posterior chamber is filled with a transparent jelly that gives the eyeball its shape, but is also optically pure, so light rays can pass through here. And we replicate that in our model here with this transparent bubble, if you will. But this is what we call the vitreous. This is what we call the vitreous humor. The lens sits right here in front of this. And so behind the lens is this great big goopy ball of jello or jelly that when we open up the eyeball, we pop it out and it sticks together. And well, we really do. And it's transparent. And it's called the vitreous humor. Now, in front of the lens is, we call this the posterior chamber. In front of the lens, we have what's known as the anterior chamber. And the anterior chamber is the area between the cornea and the lens, and it's filled with fluid. We call that the aqueous humor. Now, this fluid that we have in that region is coming from the salt water that is generated from the lacrimal gland. The lacrimal gland releases salt water, we call it tears, that we rinse our eyeballs off with. Every time we blink, we're cleansing the eyeball to get rid of dust and dirt and debris. And if there's some certain antibacterial properties of the lacrimal fluid. and then it drains away. If it doesn't drain, if it becomes trapped inside the anterior chamber in the what we call the aqueous humor, it causes pressure on the eyeball. This is what's known as glaucoma. Glaucoma occurs when we don't drain that fluid, it accumulates and gradually starts destroying the photoreceptors. And it's a very insidious little disease that causes destruction of the photoreceptors slowly, slowly, slowly. And suddenly you discover, it really is one of those things where you wake up one day and discover that I've lost half my visual field because it sneaks up on your patients. So if you've ever had, when you go in for your eye exams and you're sitting there and you have your chin in that little machine and they say, you're gonna feel a puff of air. We've all been there, got the puff of air. It's testing for glaucoma. The puff of air can measure the pressure in the anterior chamber of our eyeball. Pressure rises if we're retaining fluid in there. So it's a very serious condition. And you're not going to know you have it because you're not going to be noticing that your visual field is shrinking until about half of it's gone. And then, what do you do then? Well, this is why we go in for eye exams every year to try and detect this. So, yes.
When we get upset, why do we cry? Why, do the eyes water?
It is a sympathetic response. Apparently, not, I feel bad for you, sympathetic, but it's a sympathetic response. Your eyes water because your blood vessels are vasodilating, around the eyes. Your nose runs too, often. When you cry, your eyes water. With a sympathetic response, we get vasodilation of blood vessels. At the same time, they're going to become leaky and they're going to leak out the plasma. It's going to become interstitial fluid. Well, it can leak out all around. The lacrimal glands release more fluid. Our nose runs to try and wash away whatever kind of irritant might be there. We don't know that the irritant is emotional. We just try and wash away the irritant. So it's a good question. We've all probably experienced something like that at some point. We've all been to weddings or funerals or received good news or bad news. If you haven't, you will at some point. So, and we have an emotional response. And it's the trigger for the emotional response causes our sympathetic nervous system to kick in and say, here's what we're going to do. It's how we deal with good news and bad news. So, okay. Now, this is what the chambers look like in cross section. This big pink thing down here is the lens. Here's the cornea right here. And right behind the cornea, going up this way, you'll see here is the conjunctiva. There's the iris. There's the lens. There's the conjunctiva. Remember, the conjunctiva comes down to the cornea and stops. That's the area that gets inflamed when we have conjunctivitis. There's the ciliary body, or what's left of it here. The ciliary body are muscles that attach to the lens and can change the shape of the lens when we need to accommodate. Now, in front of the iris is the anterior chamber, or what we call the aqueous humor, because it's filled with salt water. The vitreous humor is this area behind the lens where we find this jelly-filled ball that literally does look a lot like what we have in the model. It is when you take it out of the eyeball, if you do it right, it's almost a perfect circle, a big jelly-filled circle. So, okay. Now this is what the optic nerve looks like when it's leaving the eyeball. This is the optic nerve right there. That's the blind spot. The optic nerve, we'll see, is a very rigid structure. It has the consistency of a pencil eraser. It's about the size of the eraser on a mechanical pencil, about the size and texture of that. And if you take the eyeball and push the tissue away from the optic nerve, it is a very rigid structure sticking up in the back of the eye. You'll see that next week. But there's the blind spot, no photoreceptors. The photoreceptors are here in the retina, here and here. No photoreceptors here though. There's the retina, there's the retina. You can see the layers. You can see where our bipolar neurons are located, and you can see right there, and you can see the area below that is where the rods and cones are located on the retina. But there's retina on both sides. And of course, the optic nerve itself. And it shows up very well on the optic nerve. The location of the optic nerve leaving the retina is very obvious on the inside of the retina. The layer that forms the retina is very, very thin. When someone's dissecting out an eyeball, and many times someone will accidentally scrape out the retina without realizing, what is this brown stuff, and push it aside, and that was the retina, because it is paper thin. Yeah. So what about problems with the eyes? A good portion of us have to wear corrective lenses, whether they are contacts or glasses. See, what happens when we look at we look at an object, we are seeing the light rays bouncing off that object going into our cornea. First, the cornea bends the rays a little bit. Then the lens gets those light rays and bends it a lot. And we focus, in a healthy eyeball, we focus the light on the retina. So we focus on the retina. We say that the focal point of the image is directly on the retina. So we see it sharply and clearly, unless of course we are nearsighted or farsighted. If we're nearsighted, the focal point is not on the retina, it's in front of the retina. And so if you are myopic, if you are nearsighted, which means you have great up close vision, if you're nearsighted, you're going to take a book or something and bring it in close to your face so you can see it better. Because what you're literally doing is moving the focal point of the object, which is in front of the retina, you're moving it to the retina. If you are farsighted, which is called hyperopia, you have great distance vision. You can see things way off, very good distance vision. Up close, not so much. And so a person who is farsighted, the image, the focal point of the image is not on the retina, not in front of the retina, it would be behind the retina. Now, you know the light rays can't go through the retina, so what's hitting the retina is all blurred, because if they were allowed to continue, you would see the focal point was actually behind it. So what do? They take an object and that they're looking at, and they try to pull it further away so they can see it. So, and looks something like this. There is normal vision on the upper left. Light rays are coming off the object, going through the cornea, through the lens, striking the retina, focal points on the retina. You see the image perfectly. Myopia, the image is focused directly in front of the retina instead of on it. And so what's striking the retina is a blurred image. So what you do is you try to bring it in closer. Now you can use a corrective lens to fix that. Corrective lenses are either going to be concave, they're going to be bowed in, or convex, they're going to be bowed out. If you have a convex lens, you move it towards it's the effect of bringing the object in close. If you have hyperopia, or what we call presbyopia, your focal point is behind the retina, which you can't do. What you're hitting the retina with is a blurred image. So you try, to try and correct that, you simply pull it out, or you use a convex lens to fix that problem. So most people that never had eye problems usually discover that they're gradually developing hyperopia as they age. People that have myopia develop that very early in life. both of our sons started developing myopia 5, 6 years old, and been wearing glasses ever since. But people that didn't have to wear glasses, you know, when I turned 40, I started having to, you know, I discovered I needed corrected lenses. So, and because of the lens changes its shape gradually over time. And a person who is farsighted, who has great distance vision, is focusing objects behind their retina. And so you have to accommodate for that with corrected lenses. It's also the problem that can't be fixed with laser surgery too, unfortunately. Anyway, so I mentioned color blindness. Color blindness is an inherited trait. Usually shows up in males. It's a gift from their mother, meaning it's a sex-linked trait passed on from their mother on the X chromosome. It affects males primarily, just like male pattern baldness and other gifts that keep on giving from our mothers. And the most common type is red-green color blindness, usually caused by an absence of 1 or 2 of the cones. Most people that have red-green color blindness only see things in shades of gray. That's color blindness. It's easy. There's an easy test for it. You look at, there's a series of color plates here. I'll just show you quickly. Night blindness is an absence of vitamin A, because vitamin A triggers the production of rhodopsin. Rhodopsin is the visual pigment that the rods use. No rhodopsin, no night vision, essentially. So these are the kind of plates that you would use to test for color vision. They're designed with different colors on here, usually some variation of reds and greens. A person who is colorblind, with red-green color blindness, would not be able to see the 25 on here. All they would see are shades of gray. Yeah. There's a 56, a little more, a little busier picture than the 25. But again, you're different. You're getting different shades of blues and greens and reds in here. And again, the reds might show up as dark gray and the greens might show up as light gray, if you could see it, or it all disappears. So there's one that's a little more challenging. Yeah. And we see 29, 45, and 8. And the test plate, the sample plate that they have in this book right here, which we have somewhere, it is this is the Ishihara color plate developed by a Japanese ophthalmologist, and it's become the standard. You flip through these plates, and there are some that are test plates designed that nobody sees anything, just so that you know it's not are you making this up as you go along type of thing. So, and it's pretty effective in determining, is your patient suffering from color blindness and what type. So, yeah, let's see. This is the. Let me go back. This is an eye chart. We've all seen this eye chart before somewhere. We don't use, most optometrists don't use eye charts anymore. They usually use those devices that go over your face, and they go, now this or this, and they keep switching out the corrected lens combinations. But this was the original Snellen eye chart, and this is where we refer to as 20/20 vision comes from. The Snellen eye chart was designed with the physics of the light rays coming off the image, striking the eyeball at 20 feet. And so your patient would stand 20 feet away from the eye chart, which would hang on the wall. In fact, there's one hanging on the wall over in the other classroom. And you would stand at the 20-foot line, shut one eye, and then you would read different lines on the chart. Now, the second line up from the bottom is the 20-foot line. And if you stand 20 feet away from that eye chart, the eye chart is mounted 6 feet above the floor, and the eye chart is of a specific size. It's about 2 feet by about a foot. If you can stand 20 feet away and read the second line up without corrected lenses, that's the 20-foot line and you're 20 feet away, then your vision is considered to be 20/20. You're reading at the 20-foot line what everybody reads at the 20-foot line. If you can read the line below that, which is the 15-foot line, then your vision is considered to be 20/15, meaning you can read at 20 feet, but everybody else has to get at 15 feet. 20/15 is considered good vision. On the other hand, if at 20 feet you can only read the letter E there, then your vision is considered 2200. Because you're seeing at 20 feet, but everybody else can see at 200 feet. And it works. It's not rocket science on this. It's all the work has been done ahead of time, and you can test the eye chart to see if you can read it. In fact, that's how I discovered that one of our sons needed glasses. I was working as an adjunct in Virginia, and it was the end of the semester, and I was wrapping everything up, and he was 6 years old, and I brought him in to help me put everything away. And he saw the eye chart, and he was standing there, and he couldn't read it. And that's how I found out that he needed corrective lenses. So, because he couldn't read the chart, he couldn't read the 20-foot line. And he'd gone through kindergarten and first grade because he sat in the very front row. And he was able to see things without having to sit 20 feet away. So also explains why he was playing soccer. He was a goalie and he couldn't see the ball. So he had a miserable experience playing soccer. A 6-year-old getting boom, whacked in the head all the time. Oh, I felt terrible. So not as bad as he felt, but anyway, that's the Snellen eye chart. Most optometrists and ophthalmologists will have a variation of that. When they examine your eyes, they may just use that device that allows them to put in different corrective lenses, but sometimes they'll project different lines on the wall, based on different sizes to test your visual acuity, but it still works. Now, this other test here is for astigmatism. Astigmatism occurs when your cornea is changing its shape. It's gotten thicker, and it causes the light rays, instead of focusing instead of narrowing as they go through the cornea on their way to the lens, to broaden. And so you see images as fuzzy objects. Lots of people develop astigmatism as they age. And what you see there, you'll notice that some of the lines seem very sharp and crisp, and some are thick and appear darker than the others. This is what a person with astigmatism would see when they use this test, because normally the chart has all the lines of the same thickness and the same dark intensity. But a person with astigmatism would see some as darker than others and some thicker than others because the light rays are being bent. So no, you don't have astigmatism if it looks fuzzy to you.
No, I'm looking at that there's a second 3 where I thought a 4 would be, or maybe.
There is a second 3 in there, and it's yes, you're right. So
I was wondering if the test had something to do with that.
Yeah, I don't know. I don't know why it's like that. So it's like watches. If you have Roman numerals on your watch, you don't have a Roman numeral IV for 4. You have 4. It's it's totally uncalled for, but that's what they do. I have to explain that it's only an example, because I used to just put it up on the screen and people would go, I can't see, do I have astigmatism? And, some did, but, people were getting a little excited over that. So I shouldn't have been so sarcastic. Anyway, because this is what happens with astigmatism. Lots of people have astigmatism. The cornea changes its shape, and instead of helping to focus the light rays through the iris, through the lens onto the retina, it broadens out. And so we get a fuzzy image. And we use corrected lenses for that too. Okay. And that is as far as we're going to go today. So next week, we will go through the ear and look at the ear models. So I've got this great ear model. This is my ear model. It is a great ear model.
It looks like it'd be good for running models.
I want to show everybody what the ear model. This, you can see the ear model there. This thing is huge. It's about 3 feet across. So we're going to look at this next week, and then we're going to look at the brain and the eyeball, and we will dissect some eyeballs and dissect the brain, and then we will be done with AP1 Lab. So, okay, so let me get us out of here. And in lecture on Thursday, we will continue in the nervous system.